Mauro, the floor is yours.
Thank you. Can you hear me well? Is it okay? Okay. Great. So I didn't know, so I'm the last talk of the day, right? Yeah. Okay, you guys are all tired. I'm going to exploit that. And so thank you for being here. So yesterday I gave you a very panoramic notion, I hope, of how messy a real brain can be, and the kinds of trouble we get into if you think about doing some kind of modeling, right? So I showed you this Pottians-Dismal model, which is a very detailed model, very successful numerically, and in my opinion at least, not very helpful in understanding what's going on. So today I take a completely different approach, and we'll talk about two, in principle, seemingly unrelated ideas. I will talk about two different lines of research, and just leave it at that for today. And then tomorrow I'll try to connect them somehow. And now we'll talk more about physics idea for real, not like yesterday. So since I'm going to mention some works from my lab, these are people from my lab, from other labs we collaborate with, and the people who pay for everything. And again, the overall scenario hasn't changed. As compared to this well-taken care of golf course, which we could look at as being theoretical physics, of course, that matter not shown. Theoretical neuroscience is more of a mess, much more exploratory, and people are coming up with ideas that can try to glue this microscopic information together somehow. Okay? Right, so let me start with this idea of brain criticality, dynamic range, and avalanches, which is work that I started almost 20 years ago, I think. It didn't start with me, of course. So let me start with the founding experimental paper on this subject, which was published in 2003, Journal of Neuroscience, by John Baggs and Dietmar Plants. This is a lab at the NIH. So I think you appreciate from yesterday the advancement of recording techniques from that one wire that people had to insert onto this giant squid axon in the 30s, 40s, to these multi-electrode arrays, which were state-of-the-art in the late 90s, early 2000s, okay? So this is in a Petri dish, and you put on top, this is 1 millimeter, so you have a couple of tens of hundreds of microns between those 60 electrodes. It's like 8 by 8 without the corners. And on top of this Petri dish, John Baggs and Dietmar Plants put slices of the red cortex. So it's not an animal we're talking about. It's a slice of the brain of the animal, plus some, not only cortex, but some subcortical regions like VTA, ventral tegmental area, which gives some neuromodulatory inputs to this region. So they put a slice here. They had to culture it for a while, and they just measure spontaneous activity in this system. What happens? And what they are measuring here is what I mentioned very briefly yesterday, local field potentials. So local field potential is a kind of activity that you measure, which kind of somehow integrates electrical activity going on around one of these electrodes, comprising the activity of many neurons, hundreds of thousands of neurons, okay? So, and what you see here is like the 6 electrodes in this axis, and this one is time, on the scale of seconds. And here you have this activity in each of these electrodes. And what you see is like you have some silence, nothing happens. And then all of a sudden, you have this burst of what looks like synchronized activity, like many electrodes being active at the same time. And then more silence. And then here again, everybody, not everybody, but many of them together again.
Mauro, can I ask a question?
Yeah, sure.
So the brain, the slice of the cortex Means immediately after you cut that, you put there.
Yes. So it's a very artificial system.
Okay, but then a few hours later, you would not happen anything, I guess, no? Because
this thing is, well, then there's an art to how to prepare these slices and keep them alive for a while. So there's all kinds of special solutions that you maintain this thing alive.
It could be a reaction just after death, this type of things, this type of synchronization.
Yeah, I'll convince you otherwise in the next slides, but it could. Like I mentioned yesterday, there's always this conundrum in neurophysiological experiments, in the sense that you want to get as real as possible, as comparable as possible to the real world, but in the real world, mice are fleeing cats, right? And it's a mess. So this is like as controlled as you can think of, just as lies, for better or worse. But you're right. No, you're right. So yes. So this thing which looks like some synchronized activity, if you look in finer temporal detail, it is not synchronized activity. Here again, on a larger time scale, like 1 minute, you see this silence and silence and silence and silence. But if you look in a finer detail, like a couple of milliseconds, which remember is the scale of like one spike in a neuron, here we are not measuring spikes. We're measuring activity in one electrode, which like sums up electrical activity of many neurons around in a couple of, let's say, many tens of microns across around this electrode. So there are many neurons involved. What you see here in this finer detail is that you have here, so no activity, no activity defined. That so what is a dot here? Is some activity above 3 standard deviations or something like that. Some criterion above the mean, because it's fluctuating all the time. So you have this pa, 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 and then silence again, according to some time being that you have defined properly. It's a mess. I won't go into those details, but the being matters. So, and then it stops again. So this is called a neuronal avalanche. A neuronal avalanche is what happens in between two silences. And here you can see that they are not synchronized. It's like a spatial temporal pattern that starts and ends. So it's called an avalanche in the sense that it has these separation time scales. Their duration is much less than the recurrence interval, like in this no avalanche, which is something that we Brazilians have no familiarity with whatsoever. But you can imagine that snow accumulates for months, I don't know, weeks, and then when it eventually happens, like minutes or seconds, right? So this is much faster than the interval. Okay, and then there are many ways of defining what the size S of an avalanche is. You can just, the simplest way is just count ta, 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 and see how many events happen during an avalanche. And you can also measure the duration of the avalanche, like the number of beans, or the duration in milliseconds, or whatever. You could also sum the intensity of the peaks and find that as the size of an avalanche. It doesn't really matter, okay? And then, okay, that's a very simple experimental setup. And you could ask, well, what is the typical size of an avalanche? What is this mean size plus or minus standard deviation, blah, 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 right? You could do that. Or you could be more general and just sample the probability distribution of this thing. What is the probability distribution of finding an avalanche of size S? And these are the experimental results. This is probability, this is the size of electrodes, measure this, the number of, as a number of electrodes, or the sum of the peaks, etc., and varying the spatial configuration of the multi-electrode array, which is sitting on the bottom of this Petri dish. It doesn't matter how you do it. Essentially, you always get, within limitations, of course, a power law. This is log scale. This is a straight line. It's a power law. And when they measure the exponent, physicists immediately recognize exponent. So First of all, so the distribution of avalanche size S is a power law. And it's very well fit by this exponent here, 3/2, 1.5. And if you're a student and never heard about this before, the first question that should be in your mind is probably, so what? Right? Who cares? That's always a good question to ask. And that means it's a fat tail distribution, which means that the mean size of an avalanche, or the typical size of an avalanche, or the mean plus or minus standard deviation, these are all meaningless questions because the variance of the distribution is infinite. Therefore, whatever is your mean, whatever your mean is, you have infinite variance, and therefore your system is governed by fluctuations. Yes? Yes, you can ask anything.
So you showed the graph before that looks like synchronized behavior. And we know that synchronized behavior is linked to epilepsy in the brain, more or less. But then you told me that's not synchronization, that's an avalanche. So what does an avalanche represent in the brain? Do we know?
Oh, that's the like $1 million question. We can come back to that. But it's not epilepsy because, so epilepsy, it's usually have synchronization with some periods. Tra, tra,
Yeah.
tra. It's periodic and synchronous. Here, it's not periodic,
Yeah.
and it's not exactly synchronous. It depends how, okay, so synchrony is a word that physicists use very freely without not always defining things properly. And in neuroscience, there are many, many different ways of synchrony. In physics, we are used to synchronization with periodic behavior, whereas you can have synchronization without periodic behavior. Like everybody goes together, but not periodically. That's synchronization, one kind of synchronization. There's phase synchronization, there's amplitude synchronization. So, right? So there are differences. So what I can tell you is that this kind of activity, well, first of all, this I'm trying to show that it is not synchronous. It is not.
But it is something sort of anomalous in the brain. It's not like normal, regular brain activity, or is it? That's what I'm not understanding.
I'm going to try to convince you that it is normal, regular, but still, I'll try to raise all the experimental limitations whenever I can, okay? Not my experiments, but so for the time being, this is just, the thing is that this is not a normal brain. It's a slice of an animal which is now dead. The slice is still alive, right? But the animal is gone forever. And so just like her question. So it's a very artificial system. And whatever inference you want to make from this to a real animal, you have to convince people. That's how biology starts. That's normal. Okay. So I love this kind of question. Please keep sending them, right? It's always important to repeat the caveats and the limitations. So what I was saying that this kind of distribution, which is fat tail, so these questions here, oops, sorry, are meaningless because it doesn't mean anything to talk about the mean if your variance is infinite, right? So that means your system is governed by fluctuations. This artificial one, which if people are thinking about what do the spikes mean when the brain activity is going on in the brain, it makes you think of your grandmother's chocolate cake, right? Or you can feel the smell of something and remember your childhood. Those things have, whatever happens in your brain, eventually have meaning. And people, the $1 million question is how you connect those two things. What's happening there and what you feel. We have no idea how this works, okay? But, so that's a goal. So in statistical physics jargon, this is a non-trivial scale invariant statistics. And people recognize the exponent. The physicists recognize the exponent. I'll show you. You have a much uglier power law for the duration, lifetime of an avalanche, which with some good goodwill, you recognize exponent 2 here. Of course, it's less than a decade. It's just 64 electrodes. There's not much you can do about it. This is just experimental limitation. This was 10 to 20 years ago. Now it's different today. And now, okay, now this animal is alive. Now this is anesthetized rat. Okay, it's anesthetized. Right? It is anesthetized. So it's not a performing animal. But it is an intact brain in which you insert electrodes and you got something like this. So now you have more than 1 decade in this power loss. Again with the same exponent, okay? 3/2, 1.5. Yeah, but the animal is anesthetized. Yeah, I know. Now, these monkeys are not anesthetized, okay? These monkeys are not anesthetized. And these are monkeys from Miguel Nicolelli's lab in Duke University. So these, I don't know if you know, experiments from his lab. They were very famous experiments in which you record it from premortal cortex, which control movement, and then with his brain, the monkey could control a robotic arm in the other room, right? So, except that in this case, the monkeys are well trained to be resting. So they're doing nothing. The motor cortex, which is responsible for movement, is not, so the animal is not moving. Okay, but still, you get the same power laws here, again with the same exponents, blah, blah, blah. You see there's a dependence of the exponent on the time bin. This has been known for a while. That's the kind of discussion I will not have time to go into, but yes, there's that. Okay. So the thing is that these results, the first experimental results, just raised many flags for many physicists and kind of fuel this so-called critical brain hypothesis. What is that? The critical brain hypothesis is an idea, it's a conjecture, that the brain, understood as a dynamical system of very, 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 very high dimensions, operates near a critical point. So Dante Calvo is one of the proponents of this idea back in the 90s with Perbach. Perbach, if you guys probably, I don't know if you know him, but in the 90s he was responsible for the idea of proposing the idea of self-organized criticality in many, many systems, has a book with a very modest title of How Nature Works. And there are some old reviews. Now, there are more recent reviews I can put in a different slide tomorrow. Reviews on why this idea makes sense. So some people claim that Turing somehow alluded to these ideas back in the 50s. So this is a cartoon of a second-order phase transition. And you forgive me, as physicists, I'll talk about second-order phase transitions in a very simple sense because sometimes I present these things to biologists. So yesterday I had to present very simple things to you about the brain that the biologists know. When I present this to biologists, this is the hard part when I talk about second-order phase transitions. Okay? But this is a cartoon. You have some order parameter and some control parameter. I mean, in the Ising model, it's a temperature, for instance, and then you have a qualitative change of behavior here, which is continuous for this order parameter, and this is the critical point. And if you take this idea seriously, you would ask, well, what is the control parameter of the brain? What are the control parameters of the brain? What are the order parameters of the brain, etc.? And if the brain is critical, what is the transition? Blah, blah, blah. So, just to be sure we know we're talking about, again, for the biologists, of which there are none in this room, this is the phase diagram of like a simple Ising model. So magnetization a function of temperature. If temperature is too high, you have no long-range order. Spins are flipping up and down. If you have too low temperature, below this critical temperature, then you have these domains with long-range order, and you have macroscopic magnetization. That's a magnet in your fridge. And in between, you have a critical temperature in which these domains have all possible size. There's no characteristic size for these domains, right? And if you want, if you're a biologist, you would appreciate a demonstration of that, showing that this thing is for real. This is a magnet. This is another magnet here. Don't do this at home. It won't work because the magnets in your home have higher critical temperature. You wait until it's cooled down, so it has gone from here to here, has become a magnet, and it sticks to the other guy. Okay? So this is the same thing, except with the spoiler. Now you know what's going to happen. Okay, just 10 seconds. It's nice. Oh, the guy went first. Right? So it's very high temperature, and it's cooling down. Eventually, when you reach the point, it becomes a magnet and sticks there. Right. So, cool. The question, of course, yeah.
The brain might be near a critical point, right? It's a very interesting idea, but like that might not really be a good thing also, right? Like one would imagine that you want a brain that is not that susceptible. I mean, it would sort of imply, if it was near a critical point, that that it could be susceptible to very, very intense sudden changes in behavior in a way that might not be optimal, right? I mean,
Yes.
And we need to function in the long term in a way that, you know, some input doesn't completely scramble the way that we operate, right?
Right. So, yes and no. Yes and no. So, I'm very good at hand-waving these arguments, and so I can flip them my way. But you're right. So there's only so much hand-waving can do, but I'll do it anyway. If your brain were like a magnet here, it would be very rigid, right? Very rigid. So it spins like this. So the thing is that the world outside is not like this. The lion could be coming anytime soon. You have to react. You have to be careful with the lion. And therefore, the idea is that it's very rigid. It won't be able to respond quickly or with sufficient repertoire, dynamic repertoire, to deal with that. If it's here, well, there's just a chaotic brain. I'm sorry, it won't do anything, it will be chaotic. So that's not good either. So that's my hand-waving counter-argument to your hand-waving argument, right? But you're right. There's something there. So people should be able to go beyond that. And I hope to reach some quantitative and a quantitative argument by my last lecture on Friday. This is a huge spoiler, okay? But you're right. So let's see where can we go from here. Of course, the question that should have come before that one is, why the hell would someone expect the brain to be here? Except for physicists who like to study this point because it's interesting. It gives you papers. And yes.
The RG community, the renormalization group. So, yeah, I'm seeing my area here. So, when you show the exponents and all exponents are the same, this is basically universality. So, it makes clear sense to me that this is
yes.
near the fixed point.
Yes. Thank you for the support. But I can counter-argument to that because
yeah, sure.
Power laws are just one, and by the way, this is just one power law, or two power laws, right? So life is not that easy. People come up with other ideas of how to get simple power laws in non-critical systems.
I see.
So thank you for your understanding of universality class, which will come in handy eventually, but there are limitations that when you're talking about experimental data. That's the thing.
I see.
Yes.
I'm curious to see more.
Yeah, sure. Yeah. Thanks. So why would someone think of that? Well, I just gave one reason in answering to your question, right, with the hand-waving argument. Well, it turns out that there's more than that. So there's theory that predicts that at criticality, one could have optimal transmission capacity, optimal information processing. Very nice paper in neurocomputation. Bertinger and Natschlager, sorry about my German, yeah. Largest repertoire of metastable states. You see all those domains that form in the critical temperature in the Ising model? So they survive for a time which is power law distributed, so they could serve as some kind of proxy of short-term memory, which will not happen in the noisy phase, and will not happen in the order phase, which is just too ordered, right? And optimal sensitivity to stimuli, which is something that I published with Zamiki Nooshi, and I'm going to talk about this in detail today, okay? So you can judge for yourself. So these ideas, most come from theoretical works, and you see that this thing is nice, this thing is nice, this thing is nice, this thing is nice. If I had a brain, I would love to have those things, right? All the properties are supposedly highly desirable for a brain, and therefore, natural selection, the oldest and all-governing theory of biology, would imply there will be some evolutionary pressure for it to be there. So this is a very panoramic argument. Again, with this question, if the brain is critical, what is the phase transition? Right, so if you are in a phase transition of sorts here, theoretically speaking now, you do expect, if you define avalanches properly, for a given universality class, or a given class of models, then you expect these things to be power law distributed with some exponents. Let's call it tau. And the duration of avalanches are also expected to be power law distributed with another exponent. Let's call it tau t. And when people notice that tau was 3/2, again and again and again, they immediately came up with what, sorry, I'm sorry, what quickly became some kind of standard model for neuronal avalanches, which is called the branching process, or in the jargon of statistical physics, mean field directed percolation universality class, talking about universality class. So this is a very, very, very kind of simple model, which is known, which has been studied since the late 19th century, right? The idea is that you have a bunch of neurons, they're connected to some other neurons with some probabilities, try to connect with yesterday's lecture, and suppose you have some probability connectivity here, some probability of connection. If probabilities are small, somehow, then you start some activity here, and then this thing will eventually die out, die, and then disappear. And once it disappears, game over, nothing else happens. In statistical physics, that's called an absorbing state for systems which have an absorbing state. It could be 0, 1, for instance, right? If these probabilities are larger, then eventually the things start to increase and increase and increase, and it will eventually saturate to some self-sustained value. And this is the supercritical regime for that class of models in the mean-field directed percolation universality class. So the order parameter here could be just the density of active sites, stationary density. So eventually this thing starts functioning on its own. And you have a critical point in which you are on the verge of dying and not dying, and that's a phase transition. Okay? So this model, I think originally was called the Galton-Watson model, which was proposed in the 19th century to understand why some British surnames would survive in society where others wouldn't. And guess what? People who have more kids win, right? That's it. So people who have stronger probabilities of generating offspring here, they will survive. And there's a critical number above which this happens and below which this doesn't happen. That's it. And then eventually this became the branching process. So there are different names for how this thing was evolved. Once it was put into some topology in a lattice, square lattice, linear lattice, or whatever, then becomes the contact process and becomes the directed percolation universality class. Right, so.
There's a question.
Yeah, sure.
I'm just wondering, given how non-trivial the exponents are for the directed percolation universality class, that they are irrational numbers, right?
What?
I thought that the exponents for the DP universality class are irrational numbers.
Irrational or rational?
Irrational.
Yes, for infinite dimensional, below the critical dimension. So what I mentioned here is the mean field version,
Right.
mean field hypercorrelation. So if you like him are familiar with the notion of universality class, that's above the critical dimension, which is 4 in this case, right? Yes, but in general, yes. Yes. So it depends on dimensionality, right? So this is a cartoon of what's going on if you run a simple model. I'm going to show you the model soon. The density of active sites as a function of time. If you choose one of the supercritical coupling regimes, you see you have 3 runs. You start with 10% here, and it goes to 0, goes to 0, goes to 0. There are 3 runs here. It goes to 0 very quick. If you choose 1.05, which is supercritical, it fluctuates, but it reaches some kind of stationary stable, which is a non-equilibrium stationary stable because since it has an absorbing state, it's an intrinsically non-equilibrium system. I won't go into details of that, but yes. And if you go to the critical point, which here sigma equals 1, it goes to 0, but it takes longer, and the time it takes to die out is like power law distributed. There's no characteristic time for it to die. So you have major fluctuations. So for this universality class, these are the you can calculate those analytically, those exponents. And there's a third one, which I'm going to mention now, I'm going to use in the example I'm going to give you, which is if you are at the critical point and you stimulate your system with an external system, with external input, so to speak, if you are here, you perturb the system, activity will grow like linearly with the intensity of the stimulus. If you are here, it doesn't matter what you do because it's spiking anyway. And here, you are very susceptible. You respond non-linearly with an exponent which is less than 1. And for the universality class, you can calculate it's 1/2. Right, so this is mean field direct percolation.
Sorry, can you come back just a little?
Yeah, sure.
I see the exponents for the size of the avalanche, and then you have the second one. And
For the duration of the avalanche.
okay, and then what is the third one?
The third one is this one, is the response exponent. It's like this. Well, I'm going to show several plots now, if you want, but essentially, if I make this plot of this stationary density of active sites as a function of a stimulus intensity, let's make this plot log normal, and if you are subcritical, essentially this exponent is 1. You have linear response. You put more stimulus, well, the thing responds more. You double the stimulus, you double the response. That's very easy to understand and reasonable. If you are at the critical point, however, so this is like the third axis, right? Coupling.
Okay.
This is your network activity, like spikes per second or so. And this is your, like, could be a Poisson rate with which you strike your net, right? For this guy here, it turns out that the exponent is 1/2. So it's less than 1. And I'll show you the consequences of that, particularly to your question, I think. So, please, let's try to remember what I told you yesterday about the Hodgkin-Huxley model, how painstaking that was to obtain, like a couple of decades of research in World War II and stuff like this, and then they discovered the Hodgkin-Huxley equations, and then synapses and stuff like this. And then the physicists come and reduce that thing to a cellular automaton with n states, right? So working in this field can be very challenging. You have to talk to biologists and listen to them, and because it's very easy to offend someone who works in their whole life do those equations. Say, okay, I'll say that a neuron essentially is like something which is quiescent here, stable, there's nothing. If it is properly stimulated, it goes to a spiking phase, and then becomes refractory. Remember, depolarization, repolarization, hyperpolarization, it becomes refractory, and eventually comes back here and waits again for another stimulus. So that's, It's not even a toy model. It's a cartoon model of what a neuron does. But why do we do this? Because this is simple enough that, one, I can do calculations. Two, I can simulate 10 to the 5 of them in 2004 laptop, right? Today you can run it on your cell phone or something, I don't know, on the cloud. So, and then what I'm going to do is take this kind of model and put one of each of these guys here in different sites on some topology, which I'll mention briefly, and then connect it with some probabilities. So, for instance, this guy here has 4 neighbors. I has 4 neighbors, so Ki is 4. That's like the degree, the number of connections it has. And I put some probabilities here, Pi1, Pi2, Pi3, Pi4, 0.1, 0.9, 0.2, 0.5, in such a way that that's the so-called local branching ratio of this I is the sum of the probabilities, which is 1.7, corresponds to the average number of spikes that I'll see here once I simulate this guy, right? So this is a local branching ratio. Imagine, I'll use olfaction as my favorite sense, just because it's easier, it's more applicable to the model we came up with. So suppose I have a bottle of vinegar, I open the bottle of vinegar, molecules of acetic acid goes floating in the air, eventually binds to your receptor here. And then this neuron spikes, because it has been stimulated. Now, according to this model, I'll draw random number 1, 2, 3, 4 times and see if it's less than these numbers. If it is, then this guy becomes refractory because it has spiked before, and this one will spike next because this probability is higher, though by chance this was chosen. Okay, now this becomes refractory, we can start again. Now, again, this is stimulated. Now, this guy spiked next time step. Becomes refractory here. Now this guy becomes refractory. Everybody's quiet. Again, like this. Only that guy. Again, like this. Those 2 guys. Again, etc. Right? So that's a model. It's a very simple model. And now let's see the consequences. Theoretically, for a ridiculously simple model, I'm not claiming otherwise. You have all the right to despise my model. Biologists have more right than you, but yes, but you still have the right, okay? So, and I'm going to put them in a random graph because we're modeling one kind of structure in the factory system, which is kind of messy, so random graph will be a messy model, a nice model for a messy thing. That's what the rationale back in the day. And the hidden rationale is that because if it's messy, it's mean field. If it's mean field, I can do calculations and calculation work. There's also that, okay? So, but don't worry, I'll skip all the calculations. Since making movies with this kind of topology is messy, I'll do everything in 2 dimensions. But then I come back here at the end, okay? Just because in 2 dimensions I can show things nicely. So what I'm showing here is by 50 by 50 grid, so 2,500 neurons. Each square is a neuron, and they're all quiescent right now. And I'm going to show here response function, so the average collective activity as a function of stimulus intensity. So low stimulus intensity would be someone at the back of the room opening a bottle of vinegar, and eventually I might smell something here. This is this end of the scale. Please note the log scale here, okay? And here is someone picks a bottle of vinegar and sticks your nose into it like this. So it's a very strong smell, very strong stimulus, right? And let's see what this model does. So I'm going to simulate at 10 to the minus 4 whatevers, right? The stimulus is just a model. It's just a Poisson rate. And neurons are not coupled at all. So it's a very boring system. This is what happens. One stimulus reaches your network, and then there's a spike, and then another one, and then another spike, etc. What does it mean to increase stimulus? So bring the bottle of vinegar closer to your nose. Well, this happens more often. I'm showing here the same plot. Please, it's the same plot. This is log-log scale. This is linear log scale, which is how biologists make this kind of plot, usually. Except for us, log-log will be important. So it's a log-log. This is a power law, but it's a power law with exponent 1. So it's a linear plot. It's boring. Eventually, this curve saturates because those guys have a refractory period. So very strong stimulus will lead to some saturation because eventually you cannot spike faster than the number of refractory periods that you have, right? The maximum frequency there. Okay, so this is boring. Now, let's see what coupling does. Coupling here, remember, is just a probability. In this case, I'm just connecting next neighbors, okay, with some probability. Let's see what happens with the same stimulus intensity as the previous one. Now, whenever a stimulus reaches your network, now there's some kind of small excitable wave, evanescent wave, which dies out after a while, but leads to some amplification, right? Because now, you see, the response here is larger than it was before. These excitable waves lead to amplification. And this could be good, because after all, this is the external world, vinegar. This is like your peripheral system, the first layers of your some sensory system for instance, olfaction. And this is what your brain up there, which is going to identify, oh, this is vinegar. This is what they receive is this axis, not this one. Right? So here, maybe it would be too weak for you to detect, but with some coupling, now it got stronger. So there's more chance to be detected somehow. Okay? Well, if amplification is good, then more amplification is better. Right? So if I increase the coupling, you see that those waves last longer. There is more mass. So therefore, increasing your amplification, and therefore increasing the chances that you are going to detect it somehow later on, downstream in your brain. Well, then, what should we do? We should keep increasing forever. Well, let's see if we are increasing the probability. There will be a point where we're going to reach a critical value of the coupling. And here is where the slope of this curve changes, because now you're in the critical point, right? And I'm going to use a very, very, very weak stimulus, weaker than before, so you have an idea, 17 milliseconds here, da-da-da-da-da. You see now, this wave is now on the verge of dying and not dying. It will die eventually, but it lasts longer. So it's amplifying a lot. And I will claim in a minute, not now, that these critical excitable waves, amount to optimal amplification, and I'll mean what I mean by optimal. I'll talk about that, I promise. I'll give you a measurement. Now what happens if we increase this further? If we increase this further, now this thing becomes supercritical, which means that even if I put a very weak stimulus here, what happens is that this activity now doesn't die anymore. Because it's so amplified that it's self-sustained. It goes on forever. Which means that if my nose were like that, someone opens a bottle of vinegar in the back of the room, one molecule hits my nose, and I smell vinegar forever. That's not a very good nose. That's what I mean by rigidity. It's responding very strongly and overreacting. And if I increase it even further, so supercritical excitable waves amount to too much amplification, if I increase it even further, it gets worse. It's not unlike if my job was for you to detect some small taps in the microphone. You can hear more or less. And I won't do this, but if I were to come close to approach this microphone to the loudspeaker, I would create some reverb effect with amplification, and that would easen for you to listen these small taps, the soft taps. If I come closer and closer and closer and closer, I won't do that, don't worry. What happens? Audio feedback, right? In Portuguese, microfonia. And then, because the connection is so strong, there's self-sustained activity. I can tap whatever I want during audio feedback. You won't hear anything. You will hear the audio feedback, not my taps. So this is a bad sensor. So it improves up to a point, and then it worsens. So even though I did everything in 2D to show you, the model original was made for a random graph, and then we generalized for other topologies later in a couple of papers. Doesn't matter. it's really robust result, what I want to talk about now. So we have this family of response curves. This is response, log, as a function of stimulus here. Subcritical, supercritical, supercritical, critical, supercritical. You could be even further down, further up, supercritical. And this is what you get for real. If you run the model, the dots are simulations, the lines are calculations, so it works because it's mean field. The calculations work nicely. That's nice. And for random graphs, which means that we are above the critical dimension. Random graphs have this effectively infinite topological dimension. Therefore, it's way above critical dimension. Therefore, it's mean field exponents. It turns out that this 1/2 exponent. Oh, sorry.
Sorry, I have a question. So
sure.
in these models, do you have, in spin glasses, for instance, you have different mean fields for fully connected models and for sparsely connected models.
True.
So here, do you have the same mean field theory, the mean field exponents for both?
For that kind of model, yes.
Okay.
As long as you have some, well, I'm assuming our random graph has like k equals 10, so there's one giant component, that's all. We are far above the Erdős-Rényi percolation limit, right? You have just one giant component, that's all.
Thank you.
Well, it turns out, look, that this 1/2 exponent was known almost for 100 years as well in psychophysical experiment. This is an experimental plot. This is psychophysics. This is early 20th century psychophysics. What is psychophysics? It's one of the first quantitative areas of psychology in which you put the subject in a dark room, you control the intensity of the light of the flash that you're projecting. It's a dot of light. So this is a physical measurement, okay? In decibels, luminance in decibels, in a log scale. And you ask the person to tell you, okay, how strong was this light to you? Give me a number. So he will say, oh, 17. And then increases, he will say, like, no, 55. Oh, this is a 90. No. And then you go this up and down for the given person. You take another person, do the same, normalize by the maximum so they're comparable, and make this plot. And the plot gives you 0.5 exponents perfectly, okay, experimentally. So this is 100 years old. If you do it for smell, it's close. It's 0.6 or so. Then we might be tempted to say, well, then our model is great because our model reproduces this exponent here. Yeah, only for this parameter here. For the other infinite values, it does not, right? So in physics, that would be called cheating or fine-tuning. But in biology, maybe not. Why? Because in biology, if you had a good reason to be here, then natural selection would make you go there. The question is there a good reason to be there unless to make the theoreticians happy because the experiments are the same? That doesn't count, right? So keep that in mind. I'll come back to that. If you look at spiking response for ganglion cells of the retina, so the ganglion cells are the less cells in the retina processing, and the first ones that spike and send the signal to the optic nerve that goes to the thalamus, that go to the primary visual cortex that I showed you yesterday, right? So again, this is a linear log scale, which is very unfortunate, but that's why I have undergrad students who then go and extract this data and then redo the plot with log scale, and here you have a 0.5 exponent within a couple of decades, more or less, okay? And 0.58. Remember I told you how some people do detailed conductance-based models. So this is a paper by the group of Antônio Carlos Roque and Ribeirão Preto. So he takes the rod layer of the retina, it takes the connections between the rod layers, and then there's the second layer here, I forgot what are they called, and then the amacrine layer, and then ganglion cells. So all those connections here, those synaptic connections, respect the ratios of network that you observe in real mammalian retinas, and they're connected to local models of different kinds of neurons here, which all respect their local electrophysiology, their local version of the Hodgkin-Huxley model. etc., etc., etc. So I cannot do this thing, but some people can, fortunately. So they do this very detailed model, and they measure the response of these ganglion cells here as a function of light intensity in the rod layer. And what they get is a 0.5 exponent. Again, so without much, they don't have the freedom to tune parameters, because if they were to, there are too many parameters. So they have to rely on what is available from experimental data and whatnot. Right. Now, so what hold on just 1 minute here. So what I'm showing here is the fact that apparently you run different models, take different experimental data, and the nicest fit to the data, either detailed simulations or experimental data, is what you obtain in a given model at the critical point. Okay. Now, remember, I gave you the freedom to not like my model. You're free to do it. Take this paper by the group of Dante Chialvo in Buenos Aires. So now they take, remember I told you yesterday those tractographies of the whole brain? So it's a completely different scale. It's the whole brain detailed from some humans. And then you put a model on top of these nodes and connect them according to that connectivity. Now, what you see here in the left is results of fMRI activity observed in a real brain. The first column is experimental results. So the brain keeps switching between those kinds of activity here, which is called a resting state network. There's a whole literature on that. And this is what you obtain running the model at the critical point, below some critical temperature and above some critical temperature. It kind of only works if it's critical to reproduce the data. That was 2013. In the same year, Gustavo Deco, another Argentinian, now in Barcelona, did the same thing. So you have this tractography here, you put a model, I don't remember the details of the model, and then from the model here, some structural connectivity, those nodes interact. Once they interact, you have a collected dynamics. Once you have a collected dynamics, you calculate the correlations between the different nodes and calculate what is called the functional connectivity, which two nodes are functionally connected when you have some correlation above some threshold, right? So from structural connectivity, you go up to some functional connectivity. Now, you have the functional connectivity of a real human in a real MRI machine, and you have the functional connectivity that emerges from a model which you run and then calculate the functional connectivity. And then you can compare functional connectivity of the model and functional connectivity of a real brain and come up with some idea of proximity between those two functional connectivities. So this is the fitting between empirical and simulated functional connectivity, the larger the better, as a function of some parameter of your model. And where is it best? Well, it is best at the critical point of the model. Again, in a completely different model from this one. It's a very different model from this one. So over and over, you keep getting these results, that the best fits to experimental data comes from critical models. So why? Why would you be there? So the reason is that, okay, apparently you're here in the red line for a reason. Well, what would the reason could be? So we came up with this idea in the context of a very simple concept, of a very simple toy model for sensory system, sensory processing. Yeah, sure.
In all those models that you mentioned, the control parameter is a connectivity in essence.
Is somehow, yeah.
Okay.
It could be local dynamics, right? I don't know if there is, I forgot how many weeks ago, there was another conference here on physics of life or something like this, when Enzo Tagliazucchi, another Argentinian, gave a very nice talk on using this kind of whole brain models and tweaking with the local dynamics instead of connectivity. And once the local dynamics themselves went into some bifurcation point, then collectively it works. It's another very different way of seeing what a phase transition would be. Okay, so look again at this family of response curves, and let me tell you what dynamic range is. So one of these blue curves looks like this, right? You have some saturation here, and then it grows up, and then it saturates here again. So suppose this is weak vinegar, this is even weaker vinegar. If my response curve was like this, the response to 10 to the minus 5, 10 to the minus 4 is essentially the same. If you are upstream in your brain, you cannot tell the difference between this and that because what you receive is this and this. The function is essentially non-invertible here. Of course, formally it is invertible, but there's noise here. You wouldn't know how to do it. So the definition that biologists use is the following. Okay, let's take this plateau here, and let's take this plateau here. Let's take this interval between the two plateaus and just throw away 10% here and 10% here. 10% above the lower plateau, 10% below the upper plateau, because this is, I don't like this thing, I don't like this thing. So this thing here is what I can consider invertible. It's very crude. Of course, you can do mutual information and much more fancy stuff. But you can do it, that's how they do it. I want to compare with them. It's their definition, not mine, okay? So this is dynamic range. That's a number of decades, so 1 decade, 2 decades, 2.2 decades, 22 decibels of stimulus intensity that are well coded by this function here. Okay? That's arbitrary definition. So let's see what happens to dynamic range as a function of, for this family of functions here. You have this upper bound here, which doesn't change much. I'll forget that it's changing. It doesn't really matter. And let's see what happens for supercritical coupling. So the lowest curve here is this one, right? So here you have this 10% cut to 0, and therefore, this is your dynamic range. Just look at, well, that's 19 decibels, more or less. I don't know. Then I put some coupling. What happens with the coupling? Well, it amplifies weak stimulus. So the next curve goes up, right? Which means this thing goes to the left. So dynamic range has increased because of increased coupling. Has amplification led to more sensitivity in the lower end of this stimuli. And I keep increasing the coupling, and dynamic range increases, and increases, and increases. So this is how I quantify how better my system is performing in coding for the stimulus. And you reach a critical point. You have maximum dynamic range. Why? Because if you keep increasing more, you have this self-sustained activity, which will be audio microphony, if I come close to the microphone and the loudspeaker, and therefore I have to discount for this thing here, and my dynamic range now is not so large. And it gets worse if I keep increasing the connectivity. So if I, as a function of coupling, make a plot of the dynamic range, it goes like this, like this, like this, and then like this, like this, like this. So it increases, and then it decreases. If I make a plot of one against the other, it has a peak. It has a peak right at the critical point. And therefore, if we assume that having large dynamic range, that means being able to discern large range of input stimulus is a good thing, well, then you have a reason to be here at the critical point, because there you have your maximum dynamic range. And therefore, will justify a posteriori why you have these power law behaviors in nature. Okay? Right. So, it's a random graph. What if I do with that the other kind of graph? Well, we'll try lots of graphs, okay? Hypercubic lattices, scale-free networks, detailed conductance-based retinal model I showed you, some disinhibition transition in antennal load, So it works in every case. Of course, if I go to lower dimensions, the exponent is different, and dynamic range gets even larger, okay? The lower the dimensionality of the network. Now, there's also that, together with Suzanne Kinouch and Renata Pazzini, we came up with a dynamics for the synapses. Remember I told you yesterday the synapses are changing all the time? So if we use what I referred to yesterday as short-term synaptic depression, if you spike too much, you get tired and you spike less. Well, this is enough to give a dynamics to sigma, which here is the horizontal axis as an independent, as a control parameter, and here has become a dynamic variable, which converges to 1 and to 1 and to 1 no matter where you start, just with some self-organized mechanism. Yes?
Quick question. Do you understand what the upper critical dimension is for this?
Yes,
model?
for this model, it's simple as well. No, for directed percolation, it's 4.
Okay, it's pure directed percolation.
Yeah, it's pure direct for this simple model. Because essentially, we're talking about excitatory neuro, there's only one species. If you had more than one species, then might change. Well, yeah.
Mauro, dynamic range means the analogy of this would be like where the brain works? Or
No, the amount of, for instance, if you lose your cell phone in your room at night, and you're looking for it, and there's some room, and then someone sends you a text message in the middle of the night, so nice, isn't it? Then you see, so that, you can see some light there, you find your cell phone. And if you lose your cell phone on the beach in Recife in the summer, you can find it easily. So the difference in light intensity between the two situations is like 12 orders of magnitude, right? So it would be nice to be able to operate in those very different conditions. And this is what dynamic range quantifies. Of course, there's your pupil opens and closes. There's all kinds of adaptations that are not here in the model. So this is just the effect of the collective phenomenon. Which
kind
are
of
not
elasticity of the brain somehow.
not the plasticity, but rather
Elasticity,
I miss a good word for this. But your ability to handle different stimulus intensity. So this is nice, but this is the theory, right? Again, with a very crude model. Is it true? So I was very fortunate to be able to go to a conference and meet Dietmar Plans, the guy who had done that first avalanche experiment, and some of his postdocs. And over dinner, that's why I should go to dinner, conference dinner. Over dinner, I convinced, I think, after much talking, one of their postdocs to do this. Can you do this in your lab? Test dynamic range. And they did. I don't have time to go to the details, but essentially, this, in the same cortical slices, they stimulate their network. They have something aching that compares to some of this control parameter, and they measure dynamic range. And see, the no drug condition is a normal slice, which is something like this, the black ones. And PTX is a kind of pharmacological manipulation that you do in your network that increases excitability. So it gets like supercritical. Then they move to the right. Whereas AP5, the NQX, is a kind of pharmacological manipulation, which decreases excitability, so increases inhibition. So they move to the left. And most of them are in the middle. And you're complaining that in your minds that the plots are ugly. Yes, go ahead and try to do the experiment. Come on, we're theoretical physicists. So this is very wet and dirty and complicated. And okay, it's a cortical slice. Again, we'll be back. And then years, so for the record, it just took 3 years, okay? It just took 3 years. I was amazed. I was happy for about 1 year, okay? Every day I was happy. And then, because someone bothered to do experiment, because it worked. And is it true experimentally? Again, well, with anesthetized rats, somatosensory cortex, it's uglier, but it kind of works more or less, okay? There's a similar shape. I think so. No, they have more colors here. I don't know, I don't remember the details. Anyway, this took longer. This took 9 years. Right, so what is the idea here? The idea is that this critical brain hypothesis or conjecture is kind of crazy, but gave us some insights which could be tested somehow, and some tests worked. So it looks like it might be not such a bad idea after all. And I'll leave it at that for today for that topic. Just hold that. I'll come back to it tomorrow. Now, for the second idea, I just couldn't resist. This is new in my presentations just because you invited me to the Disordered Systems Conference. And I wanted to make an explicit connection with disorder system as I know them from undergrad studies, I don't know. So, and it just turns out to be an extremely, extremely nice paper to read, which is this one. It's a paper by Bill Bialec's group in Princeton, Schneiderman and collaborators, 2006. I highly recommend it to take a read if you. It's very simple to read, beautifully written, and very nice. Let me show you what they did. So, what they did is the following. Let's start with the obvious statement that neuronal configurations are just too many to sample. What I mean is that consider n neurons, and let's reduce the state of the neuron to 0, 1, to binary neuron. Either it's spiky or it's not, right? So, n neurons have 2 to the power n configurations, right? 10 neurons have 2 to the 10, which is 1024. Okay, that looks like something I could count. But if I go to 100 neurons, which is something they can measure nowadays, well, 2 to the 100 is 10 to the 30 configurations. That doesn't look like something you can sample. No way. So, we have to rely on simpler measurements. Well, what can you do? Well, if you talk to a biologist, neuroscientist, experimental neuroscientist, once he has a measurement of this network, he'll look at the firing rates of the neurons, for instance. So this neuron is firing at so 0.2 hertz. This one is 1.7 hertz. Mean firing rates, like Poisson rates, for instance. Or you can do like 2 neuron properties, like pairwise correlations. People do study correlations. This neuron and this neuron, what's the correlation between them? Etc. Well, etc. starts to become increasingly expensive, right? 3-point correlation, well. But what could those n-square pairwise correlations tell us about those 2 to the n configurations? N here is below, n here is on the top. That's a lot of difference, right? So, this is what they did. So they had this recorded 4 neurons from the salamander retina exposed to natural movies. So again, it's a kind of artificial system, but it's the retina at least, not back there in the brain. Supposedly something very upfront in sensory processing. So I think I showed one raster plot yesterday, but it's never too much to tell you again. So this is time. Each line is a neuron. Each dot is a spike from that neuron. Okay? So 4 is already a lot. So it turns out that when they make a measurement, most pairs have weak correlations. So the probability of finding a state sigma I, sigma j is close to the product of the probabilities of sigma I, sigma j, but not equal to. Just close. Just weak correlations for most pairs. Most pairs have significant correlations, but weak correlations. It's 5, right? Okay. Okay, out of these 40 neurons, they choose 10. What time is dinner? Ooh, okay. Dinner's at 7:30, right? Okay. Right. So they look at different sets of 10 neurons, but just look at these ones. You bin the data with some characteristic time, 20 milliseconds they convince you that is the appropriate time frame in which to bin this data. And then if there's a spike here in this window, you put a dot here, otherwise not. And then from this series, you create this binary pattern. So here is 0, 0, 0, 0, everybody 0. Here you have this 1 and the third neuron. Here you have 0 again, and then 1 and the third, and 1 and the 7th neuron, okay? Etc. Have these patterns. Well, there are 10 neurons here, so there are 2 to the power 10 possible different patterns. That's okay. That's 1024. That's not too much. That's why they use it, because they can sample. Cool. So Here we're showing some 10 binary patterns out of the 2 to the 10 possible. Okay, that's very nice. Then they plot the probability of finding in the horizontal axis, the number of spiking cells in one of these 20 millisecond beans. So the probability of finding 5 spiking beans is, I don't know, 10 to the minus 2, or 5 in 10, it's smaller, and that's smaller, that's smaller. That's what you observe in the data. So, of course, the more you want, the more coincidence you want to happen in a given window, the less probable it is. And this is what you will get by just looking at the correlations. If you try to look at just at the correlations, the pairwise correlations, and try to infer this thing here, you get this dashed line, which agrees with the data, which is nice. If you assume that they are uncorrelated and try to make the prediction, what you get is the blue curve, which does not agree with the data. Okay, so apparently these correlations, however weak, turn out to be important to help describe this kind of simple data. Cool. So once you have 1,024 binary patterns, 10 binary patterns, I can count how many times each of them occurred during the one experiment. Okay? So pattern, this one here. 001, 0000100 occur, I don't know, 127 times in 2 hours. You calculate the rate per second. How many? Average rate per second. And you do this for each pattern. And here in the vertical axis, you put what will be the rate of each pattern if you assume independence. So let us go carefully here. In the horizontal axis, you have the rate. So first of all, you have 2 to the 10, 1,024 points in this plot. Each point is one pattern. In the horizontal axis, you're putting the rate at which you observe that pattern. In the vertical axis, you're putting the rate at which you expected to observe that pattern, given what you know here, assuming that they are independent. And the black line is the identity line, equality, is where you like your model to be. This is what you observe, this is what you get. So the independent model gives disastrous predictions of pattern occurrence. Okay, just to be clear, it's a massive disaster. It's very bad, because you're very far away from the identity here, from the equality. These are the one spike patterns. These are 2 spike patterns that mean spikes which have 2 ones, 1 1, 2 ones, 3 ones, etc. A kind of anti-correlation with equality, actually. It's kind of bizarre. So it fails miserably. So it points to the fact that correlation should matter, however weak. So they go ahead and say, well, we conclude that weak correlations among pairs of neurons coexist with strong correlations in the states of the population as a whole. One possible explanation is that there are specific multi-neuron correlations, whether driven by the stimulus or intrinsic to the network, which simply are not measured by looking at pairs of cells. Searching for such higher order effects presents many challenges. We will not go there. Another scenario is that small correlations among very many pairs could add up to a strong effect on the network as a whole. If correct, this will be an enormous simplification of our description of the network dynamics. Okay. How do we build, therefore I'm asking you guys, how do we build a probability distribution P of the joint sigma i's, the patterns, for the binary patterns? There are infinite number of models that are consistent with the n-square pairwise correlations. Because you have 2 to the n possibilities, you have n-square correlations. There are many models that will satisfy those n-square correlations. Of these infinite models, which one should we choose? Okay, so this is a school, right? So I think it's nice to go back in time and mention Edwin James. So this guy was a physicist who, in the 50s, published this very influential paper in the Physical Review, when the Physical Review was still just one, not A, B, C, D, E, just for the Physical Review in 1957. Information Theory and Statistical Mechanics. So please follow just the first half of the abstract because he writes beautifully again. Another guy who writes beautifully. Information theory provides a constructive criterion for setting probability distributions on the basis of partial knowledge, and leads to a type of statistical inference which is called the maximum entropy estimate. It is the least biased estimate possible on the given information. That is, it is maximally non-committal with regard to mission information. If one, he goes on, I won't, but he goes on, if one considers statistical mechanics as a form of statistical inference rather than as a physical theory, it is found that the usual computation rules, starting with the determination of the partition function, are an immediate consequence of the maximum entropy principle. So I don't know if you were taught this in your graduate course of statistical mechanics, but I argue that you should have. If you haven't, just read the paper because it's beautiful. And so, what is the idea? Suppose a quantity x can take on the values x1, xn. So it's a discrete variable, okay? And suppose we know m less than n, average values of f1 of x, f2 of x, fm of x. Average values, average values. How should we assign values to the probabilities pi, which is p of xi? This is an impossible problem. It's like if I show you a die with 6 faces, and I ask you, what is the probability of throwing my die and having a 3? What is your answer? 1/6. Why? Why? Because, why? You are assuming that they are equal. You are assuming the why are you assuming that?
No, because I have no reason to do otherwise.
Okay. Keep that in mind. Keep that question in mind. Now, okay, suppose that I told you, I could tell you, well, if I know this die, okay, and the average value is 3.5. They'll say, okay, that agrees with my theory. 1/6, that will give you 3.5. If I tell you that it's, for this die, the average value is 4.2, now you're in trouble. Now you're in trouble because 1.16 won't do it. Now what do? You do this because you have one average value, right? You have the average of the x, for instance. So what do? Know that in principle we don't have enough equations. We have, okay, for this one doesn't count, probability should be positive. Yeah, it should be normalized to 1, and you have this m constraints to pi. Right? But I have n, which is larger than m, pi's to choose. So there's not enough information. You won't solve this algebraically. Right. Just let Edwin James speak. Just as in applied statistics, the crux of a problem is often the devising of some method of sampling that avoids bias. Our problem is that of finding a probability assignment which avoids bias, while agreeing with whatever information is given that we know. The great advance provided by information theory lies in the discovery that there is a unique, unambiguous criterion for the amount of uncertainty represented by the discrete probability distribution, which agrees with our intuitive notions that a broad distribution represents more uncertainty than does a sharply picked one, and satisfies all other conditions which make it reasonable, like independent events will give rise to probabilities which are multiplied, etc. So this uncertainty here, which comes from Shannon, is the entropy, or like Edwin James calls it, the uncertainty here, minus k, which can be 1, for instance. It's kBT in theory, but it's kb it's kb in physics. Here's just minus sum over I of pi log of pi. So, I don't know, information theory has this branding problem, in my opinion, because people are talking about information, what they actually mean, how much we ignore about the system, right? If you have a coin with a, you know this plot, probably. You have a coin, the probability P of having tails. So coins is a reduced die, right? And here's probability 0, here's probability 1. This function is like this. Oops, it's a bit symmetric, like this. And it turns out that the ideal coin, the ones you should use in football matches, important things, should be right here, right? What does it mean to have maximum information? It means that maximum ignorance about what's going to happen. If I take a coin, I don't have a coin. Imagine my cell phone as a coin. So this is, I won't throw it. But so this is heads, this is tail. I bet with you, heads, you pay me $1, tails, I pay you $10. You want to play? Yeah, yeah. So okay, go like this, like this, like this, like this. 1, 1, 1, 1. So P equals 1 here, right? Information 0. There's 0 uncertainty about what's going to happen. Now, if it's 1/2, I won't throw my phone. It's here, so maximum uncertainty. So therefore, maximum information you get from each measurement you make. In that sense, it's kind of confusing, the notion information. So, James keeps on. It is now evident how to solve our problem. In making inferences on the basis of partial information, we must use that probability distribution which has maximum entropy subject to whatever is known. This is the only unbiased assignment we can make. To use any other would amount to arbitrary assumption of information, which by hypothesis we do not have. Please note the elegance of the text. People wrote so much about it back in the day. Go here and read the paper, it's fantastic. How do we maximize something like this, given these constraints? Well, we remember calculus, I don't know, 1, 2, I think it's calculus 2. You do Lagrange multipliers, right? You do Lagrange multipliers. So I won't do the whole calculation. You can find it in this 1957 paper. But the solution is this. This is the probability PI, which maximizes this given that. Please note that you have Lagrange multipliers here. The probability of finding your variable x is some exponential of minus fk of x, sum over all the functions that you know, multiplied by some Lagrange multipliers. Well, this is a problem because this is not well specified. Well, it will be. The last step is to find the multipliers by imposing the constraint that average value of k is what you know it is. And once you do this calculation, this minus del lambda log of z, where z is the normalization function, which is we call partition function, you solve for lambda, reinsert here, and problem solved. You have found a probability function which maximizes entropy and respects everything you know about the system. So what does maximum entropy mean? That means being respectful to what you know, and being mostly respectful of your ignorance. You have to respect what you don't know about the system. You know this, great. You know this, great. You don't know the rest, well, maximize your entropy, respecting what you know. That's the idea. That's all there is to it. For instance, an example. Suppose you know that the mean energy of a system is this average E, right? Well, if you know this will be like 1 Fk, well, then the probability of finding an energy E, not the average, is P of E is zeta Z of beta, 1 over Z, exponential of minus beta E, where beta here, in this case, in this scenario, is a Lagrange multiplier, which has to be obtained by satisfying this thing here. Well, I hope you recognize the canonical ensemble here, right? Please note in this slide here, beta is a temperature is Lagrange multiplier, just Lagrange multiplier. So the Boltzmann distribution maximizes the entropy given the mean energy of the system. Please, so I'm not doing this now today, but Edwin Jenny is doing his paper. He's doing, actually, you don't need to talk about ergodicity and stuff. And I'm sorry to bring this up in this conference about disorder system, where I know ergodicity is an issue, but that's what comes out of that theory. So it's inaugurated the school in physics and to the amount that information could be more important than physics. Ooh, maybe I shouldn't say this here, but okay. So this is just an example. And for just to keep the propaganda alive, there's a posthumously published book by Adam James based on some notes that he had based on 2003 was published. Very nice book, in case you want to delve further. But, okay, back to the neurons. I have to finish, right? 10 minutes, okay. Right on. So,
No. Now you have 3 minutes.
No, yeah, okay. Fine. I'm almost done. So, remember, we had this issue. How do we build a model for the binary patterns? There are infinite number of models that are consistent with n-squared pairwise correlations. Which one should we choose? Now we know the answer. The one which maximizes entropy. Well, experimentally, what is it that we know? We know n firing rate sigma I, average, and we know n-squared correlation sigma I, sigma j, average. What is the maximum entropy solution according to the recipe? Well, something that looks like this. Exponential of sum over I, hi sigma I, plus sum over I and j, jij sigma I, sigma j, which hopefully some of you will recognize. This is just, if you read something like this, the argument of the exponential being some kind of Hamiltonian, that's Hamiltonian of a spin glass with random field. And temperature 1. And temperature 1. Look, there's no beta here. The Lagrange multipliers are taken already. So it's hi and jij, right? So remember, hi and jij are Lagrange multipliers, which must be chosen such that we cover those sigma i's and those sigma I, sigma j averages. So it's kind of inverse problem. In a typical statistical mechanics problem, you are given this Hamiltonian and you expect to obtain stuff like this and see what happens with it. Here, it's the opposite, the other way around. You are given this, you are supposed to obtain those guys. It's a kind of tricky to do this, but it can be done numerically. Okay? If you do this, the JIJs and the HIs that you obtain, look at the JIJs. It's like a Gaussian-like stuff with some average around 0, but some positive negative values. It starts looking very much like classical models like Sharon, Kirkpatrick, and whatnot, right? 1970, I forgot. Something. Five, yeah, something. No, it's recorded, right? Yeah, forget about it. He said 5, yeah. Okay. So there's some local field here. So negative H values favor silence. Most of the neurons are silent. And cool. So that's my connection to disorder systems. Neurons that are described by a spin glass Hamiltonian with a random field. So that's the independent model. This is, remember, each dot here, the horizontal axis, the horizontal coordinate of the dot is the rate at which a given pattern is observed. This is 1, 0, 1, 1, 0, 0, 1, 0, etc. This is the other guy, etc. And this is the prediction from just assuming independent models. How well does the maximum entropy model fare? This model, which has been fit. So the model has only the obligation to satisfy mean firing rates and pairwise correlations, nothing else. It has no further obligations. So it has no obligation to get right all these 10 neural patterns, in principle. So these are the red dots. You get really close to what you expect. And when it gets bad, like here, it's because it's so infrequent that probably whatever you're seeing here, you're not sampling very well, because it's, right? So it works way better than it had any right to, in my opinion. Just respecting your ignorance, right? Which is what maximum entropy does. And so the maximum entropy model, based on pairwise correlation, not only reproduces this 10 neuron patterns extremely well, but it also reaches like about 90% of all information possible to obtain from the data. I'm not going to describe how they do this. I recommend you read this paper. Also a very beautiful paper to read and easy to read. So this is like the information obtained from pairwise correlation, from 2-point correlations, divided by all the information you can obtain from many point correlations. And the ratio stays like in the 90% region, therefore suggesting that this pairwise correlation, even though pairwise correlations are weak, you put them all together, you get most of the information about the system, which is rather remarkable, in my opinion. And so I talked about this, and one wonders whether there could be any connection between the two topics. So Friday I come back and we'll talk. Thank you.
Hi, thank you. So, sort of unrelated question, but I know that Gustavo Deco and his collaborators were using fluctuation dissipation theorem violations to characterize the out of equilibrium nature of the network, of the neural network. Do you know if they have checked for the criticality of the neural network using these tools as well?
No, I'm not. I don't dwell on that literature very much, so I'm not sure. Well, what I know is that Gustavo has been addressing this problem from many different angles, and he keeps getting critical points all the time. But this particular aspect of the violation of fluctuation dissipation theorem, I didn't know of, no.
Any other questions? Mauro, I must tell you that for the first time of my life, I understood what the hell is this, all that maximum entropy principle from the very basic principle.
Oh, great, thank you.
Very
Oh,
nice,
wonderful.
thanks a lot.
That's
so,
Edwin James Merritt.
Guys, see you today at 7:30 in the restaurants, and tell your friends, all the students, that they must come. Not because it's 3:30 that they need to leave. So please tell them.
And you're not telling, you're not saying that with regards to the dinner.
What do you mean?
Because it's not that they should come to the dinner.
No, the dinner I know they are going to go, but I mean, because it's for free. But this is also it's very important to come. So I've let them know. But then you all tell them that it's not possible what they did today. Okay, see you later.
Go ahead.